really is unfair to suggest that science is driven by some kind of agenda, say a naturalist agenda as opposed to supernaturalist agenda or something of that sort. Science is driven by a skeptical examination of the evidence, of the facts. So we do go where the facts lead us. You have to be open-minded to the possibility that you're wrong, and scientists actually take pride in sometimes, well, it's certainly in admitting they're wrong when they are. It's almost a matter of, I think pride is the right word, when a scientist is able to say, I was wrong, I had this theory, it's been disproved, maybe I disproved it, or maybe somebody else disproved it. That's how science proceeds. Scientists really are not out to prove something that they want to be true for emotional reasons. A scientist would never say, I want to live in a society of such and such a kind, therefore the scientific facts must go along with that scientific theory. Well, Craig, I won't embarrass you by bringing up the old Buccaneer title again, but your famous technique for analyzing DNA is called the shotgun technique.
That's right.
And that somehow seems symbolic and appropriate. When did you first think of this brilliant speeding up of the technique of analyzing the human genome?
Well, shotgun sequencing was always a part of sequencing. The biggest limitations were computational. The best software even through the 90s could only deal with 1,000 sequences. So that's why, in fact, the whole project was viewed as you have to break it down into hundreds of thousands of smaller projects and distribute them, because that's the largest you could do.
Yeah.
And it just seemed to me that the limit was just the mathematics.
So it's the computational
The
power.
computational part. And so early on, after we had hundreds of thousands of ESTs, we hired Granger Sutton, a mathematician.
What's an EST? I'm gonna.
Okay, these are the express sequence tags from mRNA from tissues that really sped up gene discovery so much.
Yes.
And so all of a sudden we had, instead of hundreds, we literally had hundreds of thousands of sequences that we needed to try put together.
The official Human Genome Project had been going for how long when you came on the scene?
Well, in fact, the first discussion started in the mid-'80s, and that's when I got actually very excited about it. I had just sequenced my first gene.
Okay, so here we've got what a much more up-to-date version of
That's right. We actually have hundreds of these machines. And massively parallelism was a key concept.
Yes.
People were looking for one machine to sequence the human genome. We just added on hundreds of them, just the same as computing, instead of one processor having thousands of processors. So in 1999, we had to build the third largest computer in the world to do the calculation.
Yes.
But computers keep growing much more rapidly.
Yes.
So that computer would not probably be in the top 200 today.
Yes. So how much faster can you do it today?
So what took 9 months at Celera, we now can do possibly in weeks.
Yes.
So we're actually in a transition phase. So all these, each one of these machines are about 4 to 5 times faster than what we had at Celera.
Right.
And then we have a couple new machines that have the potential of replacing this entire room. So it keeps changing by orders of magnitude.
Yes.
So
now,
we could sequence your genome in probably 3 weeks now.
I wish you would. Tell me about the relationship between you and the official Human Genome Project.
Well, I started out as part of that, but they didn't want to switch to try this new method that now everybody has adopted.
Yes.
Because they had the bureaucracy set up, this distribution system. And nobody ever envisioned just a small facility like this, as large as this looks for a research lab, that one center could just do it.
Quite, yes.
And not only do it in 10 years, we did it in 9 months.
Yes, yes.
And now we're trying to scale up again to go from one in a few months to 10,000 genomes over the next several years.
Yes. Yeah.
So the scale keeps changing dramatically. So we have a chance to understand the breadth of humanity at the DNA level.
Yes, and you've done your own genome, and as I learned from your book, you've learned some good things and some unpleasant things.
Yes,
as we all would if we had our
exactly.
Yes,
I argue once everybody has their genome done, will all either be equally insurable or uninsurable.
Uninsurable, yes. Yes.
So yes, we all have risk
Yes.
factors for diseases. But that's what I've tried to put in context with the book to show that, you know, there's so many things that come into play. You can have a risk for one disease and one set of genes, and another set of genes could totally compensate for that. And that ignores the other half of the equation, the environment.
Yes, sure. Which is, of course,
so we're in the process of trying to get real
yes.
data on what we can learn from the genome.
Are you doing comparative as well? Lots of other species now?
Yeah, so now literally there's been dozens and dozens of mammals done. In fact, we measured if sequencing another mammalian genome won't give us any new genes. We've absolutely saturated that part of the tree.
Okay. Yes.
It'll just give us new combinations of those genes and variants of those. And
Yes.
so that certainly helps somewhat. I mean, but we've gone back even further. With Sidney Brenner earlier this year, we published the elephant shark genome.
Right.
So it's what, 1.5 billion years
since the common ancestor.
Since the common ancestor for the earliest cartilaginous fish. The non-coding regions of the elephant shark genome.
That's not a billion.
I
got that wrong, didn't I? It's if you add up the total distance going back and then forward again.
Exactly.
It's 1.5 billion. Yes. Okay. Right.
And so what people called our junk DNA is conserved back that far at least.
Far. Yes. So we're really fleshing out the whole tree of life, the whole evolutionary tree.
Yes.
You're looking at key points. Are you influenced by evolutionary questions? We better do, say, a hagfish because that's key for particular evolutionary junction.
Right. It's wide numbers of genomes. Instead of it being a difficult issue, as a side project, we got funding to do 160 environmental genomes, and that's just sort of a side project now. So with the environmental sequencing from the source of expedition.
What do you mean by an environmental genome?
Well, an organism, for example, isolated from a high temperature vent in the ocean, just isolated from soil sample from the ocean, etc. These have been gathered around the globe.
On your ship, on your boat?
Well, no, these are actually ones that were gathered independently by the research community.
Yes.
We're trying to do the whole genomes of those organisms to put what we're finding from the shotgun sequencing of the environment into a context. Because there's such massive gene discovery that it's hard to keep it all in view.
And you're discovering new species all the time.
So each 200 miles in the ocean, up to 40,000 new species in a barrel of seawater.
Yeah. So you're sailing through the ocean, fishing up a barrel of seawater, and then what do you do with it?
Well, we just filter them through different tiny size filters to capture the microbes and then the viruses. And we bring them in here, and basically they get shotgun sequenced. So these machines right now are doing a combination of things from human, the palm tree genome, new environmental samples from around Cape of Good Hope, and even up from Alaska this summer from melting glaciers. So it's the same technology. It doesn't matter whether it's
it is
a
so
virus.
Immensely exciting. I mean, the same technology works for everything from viruses to humans to kangaroos to oak trees. The DNA is the same kind of stuff. The genetic code is the same. Darwin would have been so thrilled.
Absolutely.
I mean, if only you could have shown Darwin around this lab. He would have been bowled over with admiration and just sheer excitement at what is now possible to do.
Well, in fact, the biggest part to me of the expedition was the Galapagos.
Oh, yes.
Witnessing all the sites that he did. Basically, it's not been changed in that 150 odd years of the same species. But he was looking at the visible world and seeing how different it was. We now have the opportunity with this tool set to look at the invisible world that he could only get hints of.
Yes.
And it shows that this vast continuity from the simplest life forms to the more complex.
He, of course, emphasized diversity because that's what he saw at the whole organism level.
Right.
But you're finding the incredible similarity that there is between creatures. I mean, even bacteria. It's the same.
That's right.
It's the same machine code that's going on.
We find themes repeated, going back into the simple viruses to bacteria, plants, animals, all the way up through the human genome. And you and I have discussed this before. This group, particularly in my country, over half the population doesn't want to teach evolution. To me, it's not a theory anymore. I've looked at the genetic code of this wide diversity of species, and it's a continuum.
Yeah. Yes. Well, evolution is a fact. I mean, that's right. There's no question about that. And I'm always being asked, well, produce the evidence, and really, you're producing the best evidence of any. I mean, fossils are nice, but if we hadn't got a single fossil anywhere, the evidence from this lab alone
on its own
would be absolutely not just enough, but overwhelmingly, staggeringly enough.
No, we really struggle to keep up with it. So here's a day's run. So each one of these little plates holds 384 samples. So and then each of those tiny samples in that plate, each one of those tiny wells will give us 900 letters of genetic code.
Okay.
So that's a pretty good spectrum for the day.
Terrific. Yes. Yes.
I couldn't even count all those plates, let alone analyze the data from them. So high performance computing is one of the absolute essentials now of biology.
Yes.
Without the biggest computers in the world, we can't do what we're doing.
Yes. And your present computers, how do they rank in speed in the world?
Well, it's we're making use of so many different computers. The one we have here is roughly twice the size of what was the third largest in the world in 1999.
That was in Celera.
So this is on the order of 3 teraops. But to do these calculations, we actually we have to use the largest ones in the Department of Energy and elsewhere, just because the calculations become overwhelming. Think if we, in a few years, if we have thousands of human genomes, all the clinical information, associated phenotypes, it's going to be one of the largest calculations ever. So I can show you the computer room if you'd like to see that.
Thank you. Yes. Lead the way.
If you want, I can show you the new technology that's going to replace this entire room with.
So what you're saying is you've got one machine now that replaces that entire room we've just been walking around.
That's right.
And when's that coming on stream?
Well, it's just went into the production facility. It's behind our secret curtain right now.
Could we look behind the curtain?
Absolutely.
Oh, wonderful! Thank you.
I'll show you our battery room first.
Yes.
Just to show you the level of the power requirements for what we do. This is not part of the normal tour. People. So this is a giant UPS system. So these are all batteries, because with all these machines and the computers, if they get a power interruption, it could cost us $100,000.
Okay.
Right?
So this is backup in case the mains electricity fails.
So everything runs through this, and then we have a megawatt generator outside.
Yes.
And so the amount of power, we use $1 million worth of electricity a year here.
Right, okay. Put it to very good use.
Yeah, that's the background of science that people quite often don't see when you just look at a little machine.
Yes.
We head down here to,
Okay.
we've been, in fact, shifting things. We have 3 major computer rooms.
Yes.
So this is just one now for dealing with the, we built a lot of room for expansion, so we're just going this, this end door. But
you also farm some of your computation out to outstations as well?
We have to, just by the sheer magnitude of it. We have to, and we're talking to some of the biggest computer manufacturers about doing even bigger calculations.
Yes.
So all these blue cables come right over from the sequencing machines.
Okay.
Right into the servers. And it's all, each one of these is equivalent to a PC, basically.
Okay,
right?
so each of those tiers there is equivalent to one. So how many PC equivalents are there in this room then?
I think there's think it approaches 3,000.
3,000.
I'm not.
Okay,
Not
and
sure
they're
the
all
latest
working
number.
in parallel.
That's right.
Together.
So that we can feed in huge amounts of data
Yes.
and keep it processing in real time.
Yes.
So this facility works 24 hours a day,
Yes.
7 days a week, mostly on automation. So,
Yes.
so you can see why we can't afford the power to go down here.
Indeed not. And where do the results come out?
The results come out. We're wired over to our other facilities
Okay,
and
right.
stuff, so we can do it there. So it's
This is all room for expansion.
yes.
So you'll just get more and more.
That's
and
right.
the thing about parallel computation is you just go on adding bits and it just.
And the nice
works
thing is
in.
they keep getting smaller as well.
Yes,
So
yes,
you know, this is like the early computers, right? They
Yes.
took giant rooms.
Yes.
Now that this whole 60,000 square foot facility may be
Right.
condensed to
Yes.
a box.
Yes.
And so now we can scale up to do thousands of genomes
Yes.
instead of a giant facility
Yes.
for one. So,
Yes.
so
Yes.
technology is progressing quite rapidly.
Yes.
The other thing is storage, as you know, with 7 years ago, a terabyte of storage probably cost us at least $500,000.
Right. And now?
Now I have a little box on my desk with a terabyte for $150. So fortunately,
Yeah.
some things go in the right direction.
Yes. What did you do 7 years ago that's different from today?
So, so 7 years ago, the public effort just did a haploid genome project. So that means it was half.
So what one?
So we get
yeah.
both. We get 2 sets of chromosomes from
Yeah.
our parents. At the time, I thought it was going to be $3 to $5 billion for half. They didn't want to tell Congress there was the other half, and they thought they could infer that. At Celera, we did shotgun sequencing of 5 people, 3 women and 2 men of different ethnicities. But when we did the assembly in the computer, it was a consensus assembly.
Okay.
So a lot of the variation got subtracted out. So
Yes.
both projects
Kind of
were
average of those 5 individuals.
exactly.
Yes.
So in terms of what a human genome looks like, both projects were very flawed. And so out of that work 7 years ago, we thought we all had the same genes, and we differed
Well,
from
not-
each other.
not exactly, but-
With a 1 out of 1,000
Yeah,
variation.
Yeah.
So now from the complete genome, both sets of chromosomes, we know you and I probably differ by 1 to 2%, which a year ago, all of us were telling the world that we differ from chimps by 1.27%.
Yeah. So we underestimated.
So we underestimated all that, and that's more like
Yeah.
5 to 6%
Yeah.
now. So, 10 years ago, all these things were done by manual processes of taking a sterile toothpick and picking clones.
Mm-hmm.
And now, this is all for the Sanger sequencing. The new technology, we don't do any bacterial clones. It's absolutely incredible. It's all amplification.
Yes.
So, you can see even this, what seemed like tremendous automation for picking the colonies of E. coli, that each one of these has a different human fragment in it. With the new technology that are coming online, they skip this step altogether because it just amplifies the DNA directly. So we don't have to clone it in E. coli. And there's tremendous advantages for that because most of the gaps in the genome were from, here you can see this.
Yes. So each of the spots is a colony of bacteria.
Bacteria, and those pins shoot down and pick it, and then it puts the samples into the plates, and then you'll see it goes and cleans off and sterilizes the tips again. And so all this is all digitized. So the green ones are all colonies that are okay to pick. It'll skip these. If they're too close together, it won't pick them.
Okay. And each of these colonies of bacteria has a fragment of human genome
That's right.
in it, which is
Each one a unique fragment.
fascinating illustration of the fact that we use the same genetic code as
Exactly.
bacteria.
Yeah, and it can simply make
yes.
lots of copies of it the same way. And so it's the automation of all these steps that were considered complex
Yes.
biology not that many years ago that have taken things totally to the next scale.
But that's been superseded. That
It's
machine
now
there.
being superseded. I mean, we still do Sanger sequencing.
Yes.
The irony is, what a wonderful breakthrough Sanger made.
Yes.
My genome is probably the first and the last genome that'll be done with
to be
Sanger
done
sequencing.
Yes. Yes.
So we've had to build all these fancy robots here that actually these isolate the DNA from the bacteria and
Yes.
produce it, where we can put radioactive labels on it and things like that. So this whole thing is shrinking down to
Yes.
small boxes. But this is even much finer than what we had at Celera, where it was,
Yes.
it was
Yes.
still relatively crude back then.
Yes, yes.
But it's just the naive notions that you couldn't industrialize biology, you couldn't do it on a scale, and we're going to be at 10,000 times the scale soon.
It's amazing. What we've been talking about has been called Moore's law. Computational power multiplies itself by 1.5. Is it? No, by 2. Every 1.5 years,
Yes,
something like that.
Yeah.
I suppose there's an equivalent of Moore's law. We could call it Venter's law.
Well, in fact,
for the genome.
Exactly. I had a fun conversation with Gordon Moore, and I said sequencing is changing faster than Moore's law. So I said Venter's law trumps Moore's law. He goes, but you're absolutely dependent on high-end computing, so Moore's law wins in the end, which it does. We are totally dependent on the computers.
So sequencing genomes is equivalent at Moore's law, at least,
Yes.
which is doubling every 1.5 years.
Yes.
Which means that in 50 years, everybody can have their genome done in the doctor's office.
I'm hoping we get to that point in 5 years.
In 5 years?
Yeah, in 5 years it should be down to $1,000, which is pretty amazing.
$1,000 to have your genome
That's
done.
right.
Which means that the doctor will prescribe not a generic cure for what you've got, but the specific cure for anybody of your genome.
Exactly. They'll take that into account in assessing risk and hopefully get us to the key change in medicine. It's going to be preventative medicine by trying to overcome what some might consider our genetic destiny. So yeah, the PCR is an essential part of, this is how once we have the human DNA isolated from the bacteria, we amplify copies of it and add the fluorescent dyes to it at the same time. So it's sort of a double step. So a typical research lab would have one of these machines.
Okay. Yes. Can you tell me something about did you encounter a lot of opposition from the official Genome Project?
Yeah, I think it's certainly not one of the best episodes in scientific history, but it's the same thing that happens over and over again when there's a new radical idea introduced in science. It receives not only intellectual opposition, but it gets very emotional on people's parts. So unfortunately it resorted to a lot of name calling, I guess because they didn't really expect to have any competition.
Yes.
But the good news, and it's the same story over again through the history of science, my worst critics now all have adopted the techniques that we've developed, and that's how genomes are getting done in the world. We just seem to have to go through these painful episodes of human emotions to accept something new.
And I suppose it's fair to say that in a sense you were building on the technology that they were using. It's just that you took it a great leap forward.
No, we're all at this stage dependent on what Fred Sanger put together in terms of the initial sequencing. So all science builds on that, and it was, it was there for helping with the initial concept of doing the human genome. It ended up being doable in one small lab versus needing the entire scientific community and every major government to support it. It was done for $100 million, which is still not a trivial amount, but compared to $5 billion, that's pretty good, right? Everything's relative in life. And now we're down to well less than $1 million to do a human genome.
Yes.
So that's a pretty fast exponential
put it in dollars
drop.
Then. Yeah. How's it dropping per year? What's the sort of dollar factor of coming down per year?
It's coming down by at least 2 orders of magnitude a year, probably. So
that means 100-fold.
Yeah.
Per year.
It's changing fast. Why don't we go through here and so once the DNA is all prepped over there, it comes over to the sequencing machines. So this was the latest version after we sequenced the human genome, and these machines are several times faster than any one machine at Celera.
Yes.
We had 300 machines there, so we have 100 here. These are probably the equivalent of 1,000 to 2,000 machines at Celera.
Okay.
But the technology keeps changing even faster. So let me show you one of one of the coolest new devices we have. It's in our secret curtain room here. Maybe we can cut through here. Let's see. Yes.
Thank you.
So this is the latest instrument out of Applied Biosystems, who made these other instruments. This one box can produce as much data as that whole other room.
That is astounding.
You know,
That
so
is astounding.
this is what we're now down to. We think less than $1 million to sequence a human genome with, versus what we just published in September required certainly tens of millions of dollars. This machine actually runs for about a 10-day period. The difference is with Sanger sequencing, we have to clone an E. coli, grow up in bacteria, the human DNA, but we get really nice long sequence reads, around 900 letters per read. All these new, very fast multiplex methods, this only gives us 25 letters of genetic code per read. So they have to do this millions of times over again to get up where we can actually assemble this kind of data. But thus far, on redoing my genome, it seems to be remarkably accurate in an early stage.
So you're using the redo of your genome as a control to see...
That's right.
Yes,
Now we did it with a really accurate Sanger
Yeah,
sequencing.
yes.
It's the sort of standard that
Yes.
the world's using now for all these comparisons. And there's about a dozen new technologies out
Yes.
there. And this is just one. We have another one, 454 machine, but it doesn't have the same throughput of this. But each technology has different advantages. So probably what we'll use in the end for the 10,000, then hopefully 10 million human genomes will be a hybrid strategy.
Okay.
We might use some Sanger sequencing, some
I
of
should
these
explain,
methods.
Sanger, that's Fred Sanger, who won the Nobel Prize twice.
That's right.
First for working out how to sequence proteins,
That's right.
and then for working out how to sequence DNA, which was the start of what you've speeded up, what, millionfold?
Probably a millionfold by now.
Yes.
And yes, but we had the pleasure of, as we've gone to synthetic genomics and the minimal genome, we went back, the first virus that we made was
yes.
5X174
Yes, yeah.
that Fred Sanger and colleagues sequenced.
Yes.
And we resequenced the whole thing
Yes.
and found only 3 variants. We don't know if they're errors or
Just
continued
different.
change.
Yeah.
And it just shows a remarkable quality of work that he
Wonderful.
did.
And what was the speed up factor compared to Fred Sanger's?
Well, for I think that took several years to do that 5,000 letters of genetic code. This machine, the total output will be, I think, 1 billion letters.
Right. Yeah.
Just from one machine.
And you're going to
for
have a
one
room
run.
full of those as well?
We're going to have a room full of these as we scale
Yes,
up. So,
yeah, okay.
you know, just think, it's this whole facility multiplied by 10,000 times.
Yes. And the amazing thing is that although obviously it costs a lot of money, it costs less and less all the time.
Exactly.
And that's what's so astounding about information technologies, that the price is coming down, not up.
Yeah, these machines aren't cheap. They're like $500,000 each.
Yes.
But
when you think of what they're doing.
when you amortize that over billions of base pairs,
Yes, sure.
it gets remarkably
Yes.
cheap.
Yeah.
So these machines cost us $300,000 each, and they're already becoming the dinosaurs of
Yes. Yes.
of the field. But
Yes.
these actually have a lot of use still. You know, the environmental sequencing, where from the Sorcerer expedition, when we get samples from the environment, the Sanger reads still give us much better information, higher quality reads, because we only use one sequence for all those. So,
Yes.
but everybody thought that, you know, the Genome Project was going to take tens of thousands of scientists, and I think it was not intuitive, I guess, to most what we did, that you could break this down and do a calculation in the computer. So for the human genome, we had to do 27 million reads off these machines.
Yes.
And so when you do the pairwise comparisons, it's 27 million squared.
Right. Yeah.
And that's why we had to build such a large
Yes,
computer
yes.
to do it.
Yes.
Where's the 454 machine? Is that? It got moved over here. have to ask directions mail. Hopefully that'll come off.
Yes.
Well, look at these plates getting ready
Yes,
for
the
processing.
plates getting ready, yes. Yeah.
Thank you. Okay. Yeah, here's one of the other technologies. So
Yes.
this is the Roche system. So this box can produce as much data as this whole facility.
Oh, I see.
As well.
Yes.
So we're going to have a dozen competing technologies.
Why are
very
you doing
soon.
competing technologies? That's
Well,
interesting.
We want to get the price down as quickly as possible. So this is not just a rich man's sport.
So it's kind of deliberate Darwinism. You're actually setting up different variants and seeing which one wins.
And we're helping with the selection because we've offered a $10 million prize to whoever gets there first.
Yes, yes.
So it's really created a lot of exciting competition, and instrument manufacturers are now competing to, the only way it really benefits the public is if everybody can get their genome sequence. It's nice to read about the advances in science, but it only becomes individualized if you can look at your own and see what you're at risk for, where you can maybe do something about it.
Yes. So it's kind of Darwinian. We were talking to somebody who said that at Microsoft they also do the same kind of thing of having half a dozen different ways of doing something and letting them compete with each other. I'm fascinated that you're doing that. Of course, people shouldn't run away with the idea that when their genome is known, they're absolutely predestined. If they were, identical twins would all die on the same day as each other, which they don't. And
so I've argued, I don't know if you agree with me, I've argued that if identical twins, if you could run that experiment 1,000 times, you'd never get the same answer twice.
Yes.
Because of all the stochastic events going from 1 cell to 100 trillion. And,
yes.
Most people, all these science fiction movies, how identical twins have the same fingerprints. They don't.
They don't, no.
Because there's so many random changes that take place.
Random changes during development, yes. Even in an identical environment, you'd still get those random changes.
Exactly.
Yeah.
So that's why, that's why this, the fear, well, we're getting into the utility zone over here.
Okay.
Maybe it doesn't matter. It's, where would you like us to go?
Anywhere, but
just keep moving, huh? Evolution doesn't stand still for anybody.
Yeah.
So it's you can see we only have a handful of employees here doing this work.
So this is normal. This would be our normal personnel in a room like this.
Yeah. So automation, all these new high throughput instruments have revolutionized the whole field in 15 years. We do things every day here that were unimaginable by most people in the field even 10 years ago.
Yes. Do you set yourself up as a resource for researchers to, suppose I was an evolutionary researcher who wanted to study the origins of some invertebrate phylum or something.
Oh yeah,
could I hire you to do the genomes of certain key animals?
Well, you couldn't hire us, but you could collaborate with us. We have collaborations around the world on all the different approaches because that's what we did with Sydney Brenner with an elephant shark. The goal is to get as much diversity. I'd be interested in looking at the whole collection that we have on genome digitized in the computer now. What's missing? What key pieces are missing that you think would help the analysis?
Yeah.
Earlier this year, from the expedition, we published a paper with 6 million new genes in it, which doubled the number of known genes from science from sequencing.
Yes.
We're about to double that number again as we continue the expedition. So that just covered Halifax through the Galapagos.
Yes.
And now we have Galapagos all the way across the Pacific, around Australia, across the Indian Ocean, and back up through the South Atlantic. So just incredible diversity that we're constantly adding in.
And that's your barrels of water.
That's right.
And is it right that you don't really care very much which organisms the DNA comes from in your barrel of water?
We don't even look at them at the organism level. We just shatter them all, take the DNA, and we can reconstruct them in the computer. Then if you actually want that cell growing in your lab, you can just take a sample of water and with cell sorting, use the DNA probe to pull that out of the whole complex.
And then you can grow it and see what it is.
Then you can grow it and see what it does.
It might be an animal, it might be a bacterium, it might be,
We can tell all that from the genetic code, and we can guess what it does.
Yes.
Imagine if, the equivalent of copying Bibles if we had to do what's on these tables here manually, it would take a large team, literally a decade or so,
Yes.
to process all this information,
Yes.
And to look at the huge amount of data that comes out of it. So I've likened our assembly, the shotgun assembly, it's like solving a jigsaw puzzle.
Yes. Yes.
Yeah, you just pick up one piece and you compare it to all the other pieces until you find a match.
Yes.
And then you do that over and over in an iterative process.
Yes.
That's what our computers do.
Yes.
Only it's a jigsaw puzzle with 27 million pieces.
Right.
Pieces of DNA 900 letters long.
Yes.
So imagine, I don't know how many monk years that would be to take that 900 base pair stretch and compare it manually to 27 million other pieces.
Yes. Yeah, so
if you could get through it once, it'd probably
Yeah.
drive you insane.
Yes. I mean, what I like about your approach is the sheer audacity of it. In a sense, anyone can understand the principle of it,
Yeah.
but having the vision to say, I will actually do it, and it works. And
it was more believing in my team and the quality they could produce. It's interesting in the history of things. Had there not been these more cumbersome methods and cumbersome thinking, this would have been the standard, and people wouldn't have thought anything of it.
Yes.
Or they said, "Oh, what a great idea! Let's do it."
yes.
Because we had horses and buggies before we had automobiles.
Yes.
We had to go through a transition.
Yeah.
And that happens with new technology in every field. And so you've experienced this as well. Moving forward in science intellectually is as much unwinding the distorted thinking of the past as it is putting a clearer idea on the table.
Yes.
And that happens at every phase of science. And you add to it millions of dollars invested interest, and it gets much more complicated very quickly.
What about your other recent project, the Making New Life?
So that's sort of an outshoot of this. So for the last 15 years, we've been digitizing the genetic code. So I view biology as an analog system. And when we read the genetic code and transfer it through this process into the computers, I argue we're digitizing biology.
Right, yes.
So now we can go the other direction. So we have billions and billions of letters of genetic code, tens of millions of genes in the computer. And now we can go the other way. We can actually take that out and put sets of genes together, chemically make the analog molecule, the DNA molecule, and then boot that up in a cell. So we can actually, it's, some people have argued that, this is the next phase after Darwin, but it's still the same principles. We're just speeding up the chance of things getting together, but it's the same selection tools, the same selection process.
Yes.
If that combination doesn't yield a living cell, then that's a dead end, right?
So you can take a genome which is just written out on paper, or could be at any rate, and you can make the animal or plant.
Exactly.
Yes, and so that means you can vary it. You can make your own animal or plant, rather than just one that already exists. You can.
Well, in fact, that's exactly what we're thinking of. We now have 10 million genes in our database. We're going to have 20 million soon. I view these as the design components of the future.
Yes.
So instead of resistors and capacitors and transistors, we have methylation proteins, we have sugar metabolism genes, we can go from CO2 to methane driven by hydrogen. We have cassettes of information processes that we can reconstruct. And we're building a robot similar to the robots in the other room that can do this on a giant scale, trying to scale up to making 1 million different chromosomes a day. Some have argued that what we're doing with synthetic genomics, potentially building these entities from scratch in the lab, is the end of Darwinism.
Oh, really?
Yes.
Because we take control from now on.
Because we've taken over from here.
Yes, yes.
And but the only difference is instead of just natural selection, we're providing the selection criteria.
Yes.
But humans have been doing that for a long time.
We've been doing that for a long time. I guess you're going a little bit further than that. It's not just you're providing the selection criteria, you're also, in effect, providing the equivalent of mutation as well. Yes. Which is a big step. But,
the random combinations, I think it's more of a philosophical step than anything else. As we,
I mean, it means mutation is no longer random. It means mutation is man-made. And,
that's right.
Yes.
And we can take this almost infinite set of genes that we have and make any combination.
Well, I could imagine a lot of people finding that fairly uncomfortable, and I imagine you get a lot of flack for that.
It's actually been it's been very pleasant. Actually, I think people are excited about it.
Yeah.
It challenges views of life.
Yes.
That you can reconstruct it in the lab. That changes.
Yeah.
I think views of life and even origins of life once we can really boot something up from scratch.
Yes, I love that expression, boot it up from scratch. Yes. That's very nice.
Yeah, we use the software analogy.
Yes.
In that,
because it is software. I mean, it's the whole DNA revolution shows that life is all about software.
That's right.
And it's just like computer tape. It's just, I mean, chromosomes are just immensely long computer tapes.
It's an inert chemical, and people have this notion that they're living things. It's just a chemical. Now we can make that chemical in the lab.
Yes.
And literally, we do boot it up by putting it in a cell that gives us the chance to do that. So I argue the software now builds its own hardware.
Yes. Yep.
Because it takes it out of that system. So the response has been, I mean, people are, I think, stunned in disbelief that science has progressed what seems to be this far. To me, it's not gone far enough, fast enough.
Yeah.
But,
I mean, I think it's a thing that Darwin would have had difficulty in understanding, because he clearly, in Victorian times, had no concept of what software is, and so I think you had to be born in the computer age almost in order to appreciate.
Yeah, I can't think what the analogy
doing today.
What would the analogy be before software? It's
well, I mean, Mendelian genetics is in a sense digital, because you know that Mendel, a gene is either there or not there, and I suppose what the Watson-Crick and post-Watson-Crick view of biology is that it's digital right all the way down.
Right.
It's digital within genes as well as between genes.
I mean, what they showed us how that information could be copied biologically into the next stage of things. So
it must have been a tremendous sense of sorry, I can't open it, can I? Tremendous sense of triumph when you got the human genome.
It was a very satisfying feeling that it was a team sport.
Yes.
And I think it represents what happens at the forefront of science and is going to be necessary for the future. I mean, we're surrounded by robots built by engineers. We have some of the most important mathematics in terms of dealing with all this complex information. The teams are multidisciplinary.
Yes.
And what my teams knew, that if any one of them failed, it didn't matter whether it was team making the DNA, sequencing it, making the computers, writing the software. Any one team failed, the whole process failed. So it was a wonderful feeling for all of us when we got to the end and it all worked.
I get a tremendous feeling of uplift when I contemplate such team. I mean, it's the same with the space organization. It's the same with. Have you ever visited CERN in Geneva?
Yeah.
Again, you get. I'm almost moved to tears by the feeling that this is human cooperation at its best. This is international, from all over the world.
Physicists manage to do that better than biologists.
Well, biologists, at least you're showing that biologists can do it pretty well.
We're showing that they can do it, but it's been a struggle as we. I've argued that maybe biologists are too much driven by Darwinian evolution. It's
too competitive.
It's survival for the individual fittest versus.
Now, this is a robot, obviously doing traditional robotic things.
That's right. These robots just isolate the DNA from the bacteria.
Right.
And it was such a tedious process. We just designed these. This is our second generation one to do this. And so they just, they go through steps, and part of what we did as humans in the lab, but then we found out robots and machines could do a lot of these better processing. So
this obviously is all under computer control.
That's right.
Yes.
It was quite comical at the start of what we were doing at Celera. You know, the sequencing machines had a robotic arm, and it was not engineered very well in the original version. And the arm would sometimes go crazy and fly across the room, and so we had all kinds of startup problems.
Yes.
You can convince people what to do with employees, training them better, but here we got to teach the robots how to do it all intelligently. But you know, the next stage, these same kinds of robots is what we'll use for making the chromosomes to design what we're trying to do for energy. So
yes, what's that about? Do tell me about the energy.
So my biggest concern, talking about survival and evolution, is that we don't do something to change what we do as a people with taking carbon out of the ground, burning it, and putting it in our atmosphere. We may not survive long, or we're going to have to adapt in new ways. Some, as you know, have argued that the only two hopes for humanity is that we can go to another planet after we've totally contaminated this one.
Yes, a slash and burn philosophy.
Or we can evolve the human genome to adapt in this mess that we're making of our planet. I'm hoping there's a third alternative, that we can use modern science to come up with alternatives to burning the oil and coal, getting renewable cycles. We see these processes in the oceans, we see them everywhere. And so if we can adapt them to production scale, we can convert sugar into diesel fuel, into octane.
Using bacteria?
Just using bacteria, using engineered pathways in bacteria.
With an energy source, what, using the sun or?
So ultimately, most of the energy on this planet comes from the sun.
Yeah.
So some cells are driven, for example, Methanococcus unacei, the first autotroph that lives in these high temperatures in the bottom of the sea. Its energy source is molecular hydrogen.
Right.
And it captures CO2 from the environment, converts that into methane. That's how it makes its energy for the cell. But at the same time, it converts CO2 into methane, which if captured gives us a chance to burn that as a natural gas
So
fuel.
you're going to dispense with digging carbon out of the ground?
Absolutely. That's
what a wonderful project, and
so I never
one
imagined
in the eye for Saudi Arabia. One in the eye for Saudi Arabia.
Well, for all the oil dictators of the world, right? Interesting, we have a partner with BP that's helping us look at biological processes to, in fact, see if we can metabolize coal using bacteria and convert that into methane.
Well, Craig, you're obviously proving that real intelligent design works.
Absolutely.
But are you sometimes accused of playing God?
It's come up on a few headlines here and there, but there's a couple different versions. My best friend and colleague that's working with us, Ham Smith, the Nobel laureate, his answer to that is we don't play.
Oh, right. Yes. What's your answer?
My flippant answer is I don't play mythical characters.
Right. Yes.
But,
okay.
You know, we've actually examined this, and we had ethical reviews before any work was done. Turns out most major religions require humans to, quote, play God.
Yes.
It's not contrary to what their philosophy is. You're supposed to use knowledge to improve mankind.
Yes.
We just view, we're trying to do that. We're literally trying to see if what we can do can prevent diseases in the future, or more importantly, perhaps prevent us turning our planet into an inhospitable environment.
Yes. Jim Watson's answer to the question, are you playing God, is, well, if we don't play God, who will?
You know, we are at a point in history where science is not an option. We are at a society that is 100% dependent on modern science. And it's a complex thing. You know, in Darwin's period, and I loved visiting the same sites he did in the Galapagos, and imagine seeing these iguanas and swimming with them. And you know, it's harder today because we have to rely on our intellects and our imagination for all the things we actually can't see. You know, we're using these incredible tools to in fact characterize this microscopic world. So we can't see it with our own visual acuity. We can't smell it. You can't see CO2. you have to take the tools of science and our imagination of interpreting these tools to understand what we're doing to our worlds around us. You know, it's easy to see if you burn your house down. That's a visible effect by just adding CO2 to the atmosphere and eventually changing the weather of the whole planet. Those are difficult concepts for any of us. And so it's a question of how to use modern science effectively and have the world population understand these are real issues, because it seems unreal, because everything we're doing is you can't see it, you can't smell it, you can't taste it. You have to understand the long-term effects of the consequences of ignoring that information, whether it's developing a human disease or changing the weather of the planet with possible catastrophic results. And so they would absolutely survive in space environment.
Yes.
And
you know, that's like Darwin did with the islands.
Yes.
It's exactly the same thing, but on a space scale.
Exactly.
Yes, wonderful.
Yeah.
So you're doing experiments to see whether DNA could survive trans-space journeys.
These organisms can absolutely survive, and
Yes.
they reach an aqueous environment. They can start replicating.
They could start replicating.
And so we've
After
launched
indefinite lengths of time. I
Yes.
mean, they've just going to suspended, not even animation.
Right, right.
You don't need animation when it's DNA.
And so we've launched billions and billions of microbes into space.
Of course, yes.
And so even if Earth wasn't populated due to panspermia, other
Yes.
places will be.
Yes.
And my favorite statistic now is this 100,000 Earth and super Earth planets in our own galaxy.
Right.
And that's just our nearest neighbors. And so there's literally going to be billions of Earth-like planets.
Earth-like planets.
And if what it's starting to look like from the synthetic genomics and synthetic biology, life will pretty much happen wherever you have the right components.
You mean Earth life will be spread by panspermia?
Well, either Earth life will be spread
Or is already there.
From the same...
Well, I tell you, my bet is that however alien it is, when we find it, it'll be Darwinian life.
Absolutely.
It'll be Darwinian life.
Yeah.
That's the one thing
Yeah.
we can be sure of.
Yeah.
Yes.